This lecture is going to cover just one concept from Chapter 8, and that is the operon. The operon tends to be a fairly difficult concept for students to grasp, so I thought it might be easier to have this as a standalone video rather than part of lecture in class, so that if you were so inclined, you could re-listen to the video or stop it or rewind it so that you'd have the time to process this concept so that you understand it well for our lecture exam. You'll remember from previous lectures that the operon, which allows for the control of the transcription of a gene, represents the highest level of control available to prokaryotes in their control of gene expression. The reason we're going to spend so much time talking about the operon is because the operon is a method that is only used by prokaryotes. It's not anything that's found in the eukaryotic world. In order to talk about the way that operons work, we're going to use two specific operon models. One is the lac operon, and the other is the tryptophan operon. We'll spend a little bit more time talking about the lac operon and then have a more abbreviated discussion of the tryptophan operon. The control of genes involved in metabolism is really important for bacteria because bacteria tend to have a relatively high number of genes involved in either catabolism of metabolites for food or anabolism of metabolites for use in the cell. So catabolism, of course, meaning the breaking down of those food products in order to fuel the production of ATP, and anabolism referring to those processes within the cell which require ATP in order to make building blocks of the cell. The two examples that we're going to use today, the lac operon, involves the catabolism of lactose, a disaccharide sugar. Then we'll switch gears and move to the anabolism of tryptophan, an amino acid. First, though, let's consider the catabolism of lactose. As you can see from the upper left-hand side of the slide, the metabolism of glucose allows for a fairly rapid growth in bacterial cultures, particularly as compared to growth when lactose is consumed. Now, we can see that, you'll remember from Chapter 6, because the slope of the line where glucose is being consumed is much steeper, and so the increase of cells over time is much more quick. If you saw these two graphs, you might be tempted to think that if you grew the bacteria on glucose and lactose, that you'd end up with a line sort of in the middle of glucose and lactose where the cells are catabolizing both equally. When you actually grow cells on both glucose and lactose, though, you see something really interesting. You see initial growth growing at a rate that is equal to growth where glucose alone is being consumed. And then after a period of time where no growth occurs, you see growth occurring at a rate that is equivalent to growth with lactose alone. So it seems that the cells preferentially use all of the glucose, and then there's some sort of period of waiting, a lag time, and then the cells start to catabolize lactose. Now, the reason for that is that glucose funnels straight into the glycolytic pathways for ATP generation, while lactose, as a disaccharide, has to be broken down in order to be moved into glycolysis. So cells will choose to use glucose as long as glucose is present. That doesn't explain for us that lag time. What explains for us the lag time between consumption of glucose and consumption of lactose as an energy source is the operon. The fact is that lactose is not as commonly encountered in the environment, and so the cell chooses to transcriptionally control the production of the proteins used to catabolize lactose in a lac operon. And that lac operon is shown at the bottom of this slide. If we consider an operon generally, we want to look at the pieces of the operon from the left side of the DNA to the right side of the DNA. We will refer to those things on the left side as upstream and those things on the right side as downstream in regard to the way that our RNA polymerase moves across this section of DNA. So at the upstream end, on the left side, we have a gene signified by uppercase I, which is the regulatory gene of the operon. Downstream of that, you have the promoter region. Promoters are not unique to operons. Promoters are regions of DNA where RNA polymerase binds and starts the transcription of messenger RNA. So hopefully promoter is something that you remember from general biology. Downstream of the promoter is an operator, and the operator is unique to the operon. Downstream of that are the structural genes, in this case, LAC-Z, LAC-Y, and LAC-A, that are being controlled within the operon. So all operons contain a regulatory gene, a promoter and operator, and then the genes that are being controlled within the operon. Now, let's take that and apply it directly to the situation that is going on in those two graphs above. Before we delve into the LAC operon, I want to take a moment to just kind of consider the overall use of the LAC operon as an inducible operon, which is to have an operon which is shut off and can be turned on. So this is an example of what's called an inducible operon. The genes in an inducible operon are in the off mode because the repressor has been bound to the DNA, and then they get turned on by the presence of an effector, which inhibits the repressor and allows for the structural genes to be transcribed. We're going to see in a moment the relationship between the regulatory gene and that repressor. So hang tight. I know repressor is a new word, but for right now, I just want you to kind of get an idea of what it is to be an inducible operon. An inducible operon is normally off because the repressor is blocking RNA polymerase from transcribing the structural genes. The inducible operon can be turned on when an effector binds to and inactivates the repressor protein. So now that we have those things straight, let's look particularly at the LAC operon. Okay, at the upper left-hand side of your screen, you see the same picture that I showed you before, an operon composed of a regulatory gene, the control region, which is the promoter and the operator, and the structural genes, which are being controlled. Now, an inducible operon has a native state that follows the pathway shown on the left. In its native state, the regulatory gene is always transcribed and translated into a protein, and that protein is transcribed and translated in the on position, meaning that it can do its job. Its job is to bind to the operator region and block the transcription of the structural genes. So hopefully now that idea of repressor makes a little bit more sense, because now you see what the relationship between the regulatory gene and the repressor protein are, right? So the name regulatory gene means this is the gene that regulates the operon because it is the gene that is transcribed and translated into a repressor, which is a name given to proteins that are capable of repressing operons. So the key thing here is that the native state of that repressor protein is to be active in the on position, and so the native state of the operon is to be turned off. In other words, it needs to be induced. And it is induced by a molecule that interacts with the repressor protein at an allosteric site. Remember, I said allosteric sites were going to be important, and that we were going to see again the post-translational control of proteins and enzymes because of molecules interacting with those allosteric sites. So when a cell consumes lactose, lactose acts as an effector of this operon by binding the allosteric site of the repressor. So effectors bind the allosteric sites of repressors. In the case of an inducible operon, the effector binds to the repressor and inactivates it. And so we call that effector an inducer because it is inducing the operon by shutting off the repressor. So when the repressor's active site has changed shape, it's no longer able to bind to the operator region, which means that RNA polymerase can move through the operator and start transcribing and translating those LAC-Z, LAC-Y, and LAC-A genes into the proteins shown here, beta-galactosidase, permease, and transacetylase, which are all involved in the catabolism of lactose. So the effector effects the operon to induce it, right? So its affected state is to be induced, its native state is to be repressed. So it's normally off, it gets turned on by the effector. This, as I mentioned, is very common in catabolic pathways. And the really beautiful thing about this operon is that it's all controlled by the presence of lactose. So if you ask the cell, when do you need to make the enzymes to catabolize lactose, the cell would say when lactose is present. So what is the very thing that regulates whether or not those enzymes are being made? The presence of lactose. And if you ask the cell a second question, when do you not need to produce the enzymes for lactose catabolism? And the cell would say, when I don't have any lactose. And lo and behold, when there is no lactose, the lac operon is shut off. Now, operons, like lots of things in biology, are much easier to understand when you see those things happening dynamically. So I want to take a moment to show you a video where you can see this control of the lac operon happening.
The lac operon of E. coli includes a promoter, an operator, and 3 structural genes encoding proteins involved in the catabolism of lactose. The lac operon is an inducible operon, meaning that the genes are usually not transcribed. When lactose is available to the cell, the operon is induced or stimulated to produce the proteins of lactose catabolism. The E. coli chromosome contains a regulatory gene near the lac operon. This gene is constantly transcribed and translated to produce repressor protein. The repressor protein binds to the operator DNA at the lac operator, physically preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for lactose catabolism cannot be transcribed into mRNA. When lactose is present in the cell, some lactose molecules are converted to allolactose. Allolactose binds to and inactivates the repressor proteins, preventing them from binding to the operator. The structural genes are transcribed, resulting in the production of the enzymes needed to catabolize lactose. Since allolactose induces or stimulates transcription of the lac operon, it is called an inducer. Notice that the inducer acts by turning off the repressor, not by actively stimulating the genes to be transcribed. When lactose is not available, the repressor proteins are active. The structural genes of the lac operon are not transcribed. This control prevents the cell from producing the proteins for lactose catabolism when lactose is not available.
All right, so that is an inducible operon. Now, one thing I want to say about that before I move to what you see in front of you is you do not need to worry about the difference between lactose and allolactose. Just lactose is the effector, and that affects the operon by binding to the repressor and turning the repressor off so that the operon turns on. Now we'll switch gears and we'll start talking about the opposite, the repressible operon. So if an inducible operon has to be induced, a repressible operon has to be repressed or shut off. So in this situation, the effector molecule is going to have a different effect on the repressor. Notice the repressible operon includes the genes in the on mode without the repressor bound to the DNA because the native state of the repressor is to be off. The effector binds to the allosteric site of the repressor protein, and that turns it on and allows that active site to bind to the operator, which represses or turns off the operon. So now, just like we did with lactose, we're going to take a specific look at a repressible operon in the tryptophan operon, which produces enzymes which build tryptophan, right? So inducible operons are for breaking things down for energy, and repressible operons are for building things that require energy. Okay, so let's take a look at the trip operon. Okay, let's take a look at this repressible operon, the trip operon. But first, let's remind ourselves of the components of the operon. Up at the top left, we have the regulatory gene I, the promoter P, the operator O, and then the structural genes which are specific to this operon, the trip E, D, C, B, and A genes. So this operon, unlike the inducible operon, in its native state, allows for the transcription and translation of those tryptophan enzymes because the transcription and translation of the regulatory gene into the repressor protein produces an inactive protein, a protein which, without something binding its allosteric site, is not active. And because it's not active, it's not able to do its job, which is to bind to the operator region of the operon. Because it can't bind, it does not block RNA polymerase, and RNA polymerase moves through the operator portion of the operon and transcribes and translates the genes for tryptophan synthesis. Just like the lac operon, though, that tryptophan is what is enabling the cell to know whether or not it needs to do this process. If you don't have tryptophan, you need to make tryptophan, which means this needs to be on. If you have a bunch of tryptophan, then you don't need to make any tryptophan, because remember from chapter 6, making amino acids and making proteins is expensive business. So you don't want to be doing this unless you actually need tryptophan. So imagine in a cell, typically tryptophan is picked up by transfer RNAs and brought over to the ribosome in order to make proteins. If you don't need tryptophan, then not all of the tryptophan will be grabbed by transfer RNAs, and some will be available to bind to the allosteric site of the repressor protein. That is shown on the right. When the tryptophan binds to the repressor protein, it affects the repressor and turns it on. Once it's on, that affected repressor is able to bind to the operator region and block the transcription of the tryptophan synthesis genes. So in this case, we call the effector the co-repressor because it is involved in repressing the operon by working with the repressor protein. So this kind of operon, a repressible operon, in its native state is induced, it's on. In its affected state, when the effector is present, the operon is repressed. So this is on and needs to be turned off. So repressible operons need to be repressed. And again, you ask the cell, when do you need to make tryptophan? When I don't have any. And when don't you? When I do. So let's again take a look at a video that'll allow you to see this process happening dynamically, because I think that will enable you to understand this process better.
The TRP operon of E. coli includes a promoter, an operator, and 5 structural genes encoding proteins involved in the biosynthesis of tryptophan. The TRP operon is a repressible operon, meaning that the genes are usually transcribed. When excess tryptophan is available to the cell, the operon is repressed, ending the production of the proteins of tryptophan biosynthesis. The E. coli chromosome contains a regulatory gene near the TRP operon. This gene is constantly transcribed and translated to produce repressor proteins. However, these repressor proteins are inactive unless excess tryptophan is available, Once activated by tryptophan, the repressor proteins can bind to the TRP operator, preventing RNA polymerase from moving beyond the promoter. When a repressor protein is bound, the genes for tryptophan biosynthesis cannot be transcribed into mRNA. When more tryptophan is available to the cell than the cell can use, some tryptophan molecules bind to and activate the repressor proteins. The repressor proteins bind to the operator, preventing the transcription of the structural genes that encode the proteins of tryptophan biosynthesis. This regulation prevents the cell from producing excess tryptophan when tryptophan is already available. When excess tryptophan is not available, the repressor proteins are inactive. The structural genes of the TRP operon are transcribed and translated, producing the proteins necessary for tryptophan biosynthesis.
All right, well, hopefully those two examples help you to understand both inducible and repressible operons better.